positions over several years, and then finally ended up in my lab. And so very happy to have her be defending the oral portion of her thesis. And then after this, her committee will meet and talk about the oral portion, and then later in the summer, we'll receive the written portion of her thesis. All right.
Thank you. Oh, Hannah, let me get that laser pointer from you. Okay. Hi, everyone. Thank you so much for being here. I really appreciate it. This is a bigger crowd than I was expecting. So kind of you all to show up. I know that the arrangement of the room is a little unorthodox. If at any point you can't see the screen or you can't see or hear me, please feel free to move your chair to a better position. No need to be at the table unless you're going to be taking notes for questions or anything like that. But yeah, feel free to move if this where you're at isn't working for you. This is, as Jeff said, my thesis defense. It is on avian malaria and its impacts on native forest birds on the island of Kaua'i. We looked at prevalence and survival as a function of time, sex, species, and family. How do we clear that? I don't want to show chat previews. Does that just stay up now that people put something in the chat? No. It won't? Okay. Cool. So just a little table of contents to give you guys a preview of what we're going to be looking at today. So for the introduction, I will give you some background on avian malaria in general. Then we'll talk about mosquitoes as a vector for that and how their distribution and infection patterns are changing with climate change. I'll introduce you to my 4 study species, and then we'll talk about the hypotheses that I developed prior to actually starting the research portion. For materials and methods, first I'll introduce you to our field sites before diving into the field methods, so our sample collection and resites. After that, we'll do a brief look at the lab methods followed by my statistical analyses. Results is going to be broken up into prevalence and survival. And then for the discussion, in addition to overall implications and impact of the findings, we'll talk about future research as well as overall project conclusions. You know how to make that go away? Hide floating meeting controls. Those look like floating meeting controls. There it is. Beautiful. Thank you, Jeff. There we go. Cool. So Hawaii, like a lot of islands, is a hotspot of both biodiversity and extinction. Up here we have a really beautiful illustration of a whole bunch of Hawaiian honeycreepers. They're a particularly stunning example of adaptive radiation, which is what happens when a species, for one reason or another, finds itself in an environment where a lot of the ecological roles are left unfilled. That species then evolves very rapidly into a lot of different species, which take on those ecological roles. This happens a lot in isolation, like islands, but unfortunately that isolation does make these species particularly vulnerable to introduced threats. On the islands of Hawaii, probably one of the biggest introduced threats is the introduction of mosquitoes, brought over unintentionally by white settlers in the early 1800s. The mosquitoes, combined with non-native birds being brought over by the same white settlers, created an environment where the spread of disease was facilitated from non-native species with resistance to those diseases to native species with none. In this case, we're talking about avian malaria, which has caused really catastrophic losses and in some cases extinctions for many of Hawaii's native bird species, with mortality rates from a single infected mosquito bite above 90% in some cases. What is avian malaria? It's a common mosquito-transmitted parasite caused by a protozoan parasite in the Plasmodium genus. There are over 40 species. They're found all around the world, everywhere except the Antarctic. And it's a pretty well studied and well-known disease in regions of the world and in populations where the parasite has a long-standing evolutionary history with its hosts. You tend to see high prevalence rates. So I have some of these numbers up here from other studies, especially from Europe. You got 91% prevalence in great tits, 50 to 85 in house sparrows, 61% hawfinches. So again, in populations that have that established evolutionary relationship, you see high prevalence of the disease and relatively low mortality. However, when there is a particularly acute infection, it can cause anemia, lethargy, anorexia, a decline in core body temperature, and hematocrit decline upwards of 50%. So hematocrit is the percentage of your blood that's made up by the red blood cells. That's essentially a red blood cell decline upwards of 50%. And perhaps for obvious reasons, birds with these symptoms, with these acute infections, don't usually get caught in mist nets. They're not partaking in their normal flying foraging activities that would lead to them flying into a mist net. Plasmodium relictum is the species causing avian malaria on the Hawaiian Islands, and it's the one that we're going to be talking about today. It's one of the most widely distributed Plasmodium species. It's really a global generalist, both in terms of host species and distribution. It's found from the Nearctic all the way down to Australia, and it occurs in at least 70 different avian families. Hawaii's avian malaria is caused by just one single strain of Plasmodium relictum. That's called GRW4. Interestingly, this is also the dominant strain on several other remote islands, including Bermuda. And we now know that this was introduced by non-native species rather than migratory birds that stop over on the island. Anecdotal evidence of infection started showing up as early as 1902, and then the first confirmed detections on the island started popping up in the 1940s. This has been really disastrous for a lot of native species. Again, we'll go back to that single bite mortality rate for Hawaii amakihi of 65%, 90% in EEV, and then in the Maui alauahio, it's 75%. These are really, really high numbers. So meet our vector. This is the southern house mosquito. It is the carrier of Plasmodium relictum. This is what is facilitating spread of infection from one bird to the next. It's a tropical and subtropical species. That means that it's really highly dependent on warm climate and standing water for survival and reproduction. Requires mean temperatures of at least 21 degrees Celsius. And if these conditions aren't met, pretty much every single measure of reproduction, development, and survival starts to see a negative impact. Plasmodium relictum, or our parasite that causes avian malaria, also does face similar thermal restrictions. Because this species is so closely tied to temperature, climate change, and an overall average global trend upwards in temperature is causing distribution patterns to shift and to expand. Now, I really love this figure. It's not mine, and it looks at a different species of mosquito, but I think it does a good job of demonstrating the overall pattern that we're talking about. So you can see that as time goes on with climate change, mosquitoes are pushing into territories which previously had been too cold for them to survive. So what does that look like on the Hawaiian Islands? So the thermal requirements of the southern house mosquito and Plasmodium relictum creates an elevational barrier. I refer to this elevational barrier as the mosquito line due to its similarity with the more widely known concept of tree line being the elevation at which trees can no longer survive, thrive, and reproduce. Mosquito line is the line at which, the elevational line, sorry, at which mosquitoes can no longer survive, thrive, and reproduce. And in 2010 on the Hawaiian Islands, this elevational barrier was measured to be at about 1,500 meters in elevation. So this creates high elevation pockets. Where is my, there it is. Oh, it goes away on the screen. Too bad. I'll just use my mouse. This creates high elevation pockets of malaria-free habitat above that mosquito line. However, climate change has long been predicted to change the elevation at which mosquitoes can thrive, pushing that mosquito line upslope and causing these refuges of habitat to become an ever shrinking pool for our endangered species that are vulnerable to the disease to inhabit. This is a problem because Kaua'i at its highest point is only 1,598 meters tall. It's only 98 meters high than that 2010 estimate. And unfortunately, the predicted changes have been occurring, and now mosquitoes are found at all elevations on the island of Kaua'i. I'd like you to meet Kauai's native forest bird community. These are the native forest birds that can be found on the island of Kaua'i. And these are the ones that we are going to be focusing on today. Starting from top left, you have Anianiao. Then down here, you have Akikiki, this is Kaua'i Elapaio, and Puaiohi. And I will introduce you to them now. This is the Akikiki. It's as cute as its name. This is one of our 2 species of honey creeper. It is a small insectivorous honey creeper that forages for invertebrates and small insects under the bark of trees, generally in pairs or in family groups. Historically, its range covered all elevations on the island of Kaua'i, but unfortunately, it's now found only above 1,100 meters in elevation. They were federally listed under the Endangered Species Act in 2010, and recent evidence suggests that the species is on the brink of becoming functionally extinct. Next up, we have Anianiao. This is our second honey creeper species. It is a small dimorphic honey creeper. Dimorphic means that males and females look different. The one that I have drawn here is a male. And they are the smallest of the extant Hawaiian birds, extant being the opposite of extinct, meaning that the species is still around. They're primarily nectar eaters. They do also eat small invertebrates. And again, they were formerly found across Kaua'i, but are now confined to just about 15% of their historic range. Next up, we have the Kaua'i Elapaio. This is not a honey creeper. This is a small species flycatcher. It's closely related to other species of Elapaio found on the islands of Hawaii and Oahu. They're largely insectivorous, and they have been noted specifically for the versatility of their foraging habits, which is a bit of an advantage as they've been seen foraging in disturbed habitat as well as intact native habitat. Historically, they're distributed across the island, but now they're rarely seen below 900 meters in elevation, due largely to habitat destruction. They are Kauai's only native forest bird currently not in decline. And finally, we have the Puaiohi. Again, this is not one of our 2 honey creeper species. This is a thrush endemic to the island of Kauai. They're primarily frugivorous or fruit eaters. They will also eat small invertebrates. Not as much is known about their historic range as with the other 3. However, the consensus is that the range has significantly contracted, and they're no longer found at elevations below 1,050 meters. Previous research tells us that this species has an ability to survive avian malaria and even maintain a chronic infection. Unfortunately, they are still highly endangered, and a 2015 survey put their population estimate at about 500 individuals. So, my 4 hypotheses. First, we expected to find an increased prevalence of avian malaria infection over time due to changing habitat and climate conditions. We also expected to find different prevalence for male and female birds due to differences in breeding and nesting behavior. Basically, during the breeding season, females are a lot more sedentary than males are. They're sitting on their nests. This theoretically might make them an easier target for mosquitoes. We hypothesized that we would find a lower survival from avian malaria in honey creepers than in native birds belonging to other families. And finally, we hypothesized a lower prevalence in honey creepers than in native birds of other families due to higher mortality from the disease in honey creepers. So for the methods, I'd like to start out with our study sites. All of the sites that you see here are sprinkled across the Kaua'i Plateau, which is, if you remember, what was that last high altitude refuge for some of the most vulnerable species. We have 3 primary sites, Halepaka'i, Kauai Koi, and Mohihi. The vast majority of our samples came from one of these sites. We had a total of 714 blood samples taken. And these sites range anywhere from 1,125 to 1,303 meters in elevation. Our secondary sites, we have Upper Kaua'i, Kaua'i State Park, and the Alakai Swamp Trail. Between these three, we had a total of 20 blood samples taken. 31 of our blood samples were taken from unknown or unrecorded locations, and 4 were taken from incidental sites that are not considered banding sites. These sites have a habitat that's made largely of mixed montane and mesic forests, dominated by 2 native species of tree, ohia and koa. For our field methods, we have our mist netting. So for anyone who's not familiar, mist nets that we use to catch the birds, you can basically think of a big volleyball net, except the filaments of the net are very, very fine. The birds have a hard time seeing it and run into it. The mist nets were checked every 20 to 30 minutes, more often in poor weather conditions. Endangered birds or those that showed sign of stress were prioritized and processed first. And in any extremes of rain, wind, or hot or cold temperatures, the nets were closed and were not used. All individuals were weighed, aged, and sexed when possible prior to the taking of that blood sample and the application of color and number bands. This number is red because I should have corrected it, one last little error. That should be that we have a total of 769 blood samples taken. And of those 769, my apologies, a total of 395 belong to one of our species of interest. All breeding birds were released back at the site of initial capture rather than at the banding station to help them maintain their breeding territories. Resites were made year-round during all field duties, and peak season for field work is from March to June every single year. Color bands sighted in the field were checked against the banding data set for accuracy before being entered into the digital resite data sheet. We had a total of 659 resites across all species. Of those, 616 belong to one of our 4 species of interest. For our lab methods, we're going to start with the malaria diagnostics. So we measured parasite load using whole blood samples and quantitative PCR. Genomic DNA was extracted with the QIAGEN DNeasy tissue extraction kit and normalized to a concentration of 2 nanograms of DNA per microliter. Samples were run in triplicate and with positive and negative controls. And the threshold of each sample was calculated as a mean of the 3 samples. This image up here, neither of these are mine, but they are in here to demonstrate this is a cell that has been infected with Plasmodium relictum. And then this down here is a qPCR amplification plot. It's how we tell how infected a bird is with the parasite. This bird that is the red line over here would have the highest parasite load, and then purple down here would have the lowest. For sex identification, again, DNA was extracted with a QIAGEN DNeasy blood and tissue kit following manufacturer instructions. And post-amplification samples were examined using gel electrophoresis. For anyone here who's not familiar with what that is, I'm going to direct you to this little diagram up here. So DNA has a partial negative charge. You put it into these wells in a block of gel and run a current through it. Because the DNA has a negative charge, it gets pulled through the gel towards the positive end, and smaller DNA fragments have an easier time moving through the gel than larger ones do, and so they move farther. What you wind up with is a spectrum with the largest DNA samples at the back towards the negative end, and the smallest DNA samples all the way forward nearest the positive end. Here in the black and white, I have an image of one of my gels that I used to sex the birds. Male samples show 1 band here, and females show 2. So we have male and that's a female. So statistical analysis, we're going to start out with prevalence. We wanted to analyze prevalence of avian malaria infection in response to sex, year, species, and family. Family being, are you a honey creeper or are you something else? 2 categories. We use generalized linear modeling in base R as well as the LME4 package to create and compare logistic regression models. Sex was investigated both alone and as a random effect and an interaction in conjunction with other variables. Otherwise, all variables were treated as a fixed effect. We were unable to model interactions between year, species, and family due to limited data and in some cases, collinearity. Our survival analysis was run using program MARC, which is a statistical analysis program specifically meant for MARC recapture, or in our case, MARC resite studies. And we use the Cormack-Jolly-Sieber live recapture model. We had to confine our analysis to the years 2012 through 2020 due to extreme variation in yearly resite effort from 2008 to 2011 that made these years poorly suited for our study. From there, we divided each year into quarterly sampling periods, and I found that the recapture probability varied by quarter, but not by year. So for any given bird, the recapture likelihood was the same in 2015 as it was in 2016. However, that likelihood was different in January than it was in July. All birds were divided into one of 4 groups based on 2 variables, infected versus uninfected, and honey creeper versus other. We would have loved to divide it all the way down to the 4 species, but unfortunately, a really small pool of infected honey creepers prevented us from doing so. A total of 8 individuals changed infection status from negative to positive over the course of the encounter history. We have no record of any birds going from positive to negative, so clearing the infection. Because we had to categorize these as infected or uninfected for our analysis, we chose to categorize these as infected, and we just started their encounter history at the first date of their positive malaria test. So sex, all the different ways that it was analyzed alone, as a random effect, interacting with other variables, did not prove to be a significant predictor of the prevalence of avian malaria. This is actually, this figure here is just the raw proportions pulled straight out of the data. You can see the proportions are very, very similar. There's a really minuscule difference between male and females. And when we modeled this, it didn't matter how we modeled it. There's no statistical significance between these two. They're essentially one group. Year also was not significant, but it's important to note that our analysis was limited by a really high variation in sample size. You can see that clearly if you look at 2016, which does not have an error bar because there was just one single data point for the year. Removal of years that had small sample sizes, including 2016, did not cause year to become significant. And our results were further confounded by sample composition. So when you have really small sample sizes like this, you're likely to run into some skew in your sample composition. And we did find that to be the case here. So of the 10 years represented here, 2 of them were missing 1 or more of the 4 species entirely, and another 6 had at least 1 species highly underrepresented, which I chose to define as making up less than 10% of the overall sample pool. Species was a significant predictor. We got a good p-value for all 4 of these at less than 0.05. So you have your, moving from here that way, your Akikiki and Anianiao, those are your 2 honey creepers, and then Kaua'i elapaio and Puaiohi, the 2 species that are not honey creepers. I want to point out that the 2 honey creeper species do have a significantly lower prevalence, both of them being below 10%, while the 2 non-honey creepers are noticeably higher, particularly the Kaua'i elapaio, which is sitting all the way up at 30%. This difference becomes even more pronounced when you group them by family. So we've got our honey creepers down here with 8.9% prevalence, and then our non-honey creepers sitting up at 25.7 prevalence. This also increased the significance. Our p-values were below 0.001, which is highly statistically significant. And I'd also like to note that there's no overlap in these 95% confidence intervals. Something that I want to point out that might be slightly counterintuitive is that the lower prevalence for our honey creepers does not indicate that these species are handling the disease well. It doesn't mean that they're not getting it or that they're clearing the infection. Rather, we believe that this indicates that these birds have a really high and really fast mortality rate from the disease. So when these birds in this category get sick with avian malaria, they just die too quickly to be caught in our nets. To wrap up prevalence, here's a table of prevalence for every single species of bird that was caught, even if it was just one. So for our survival analysis, the highest rank model compared infected honey creepers to all others. So that's infected honey creepers versus uninfected honey creepers and both infected and uninfected non-honey creepers. You'll note that the infected honey creepers here have about a 25% apparent annual survival, while everything else has about a 72% annual survival. While this is a pretty big difference, I do need to note that the small sample size of infected honey creepers caused this wide standard error that you're seeing here, along with a very wide confidence interval. So what does this all mean? We found, first of all, that akikiki and anianiao are significantly more vulnerable to the effects of avian malaria infection than are puaiohi and Kaua'i elapaio. Previous studies have already told us that puaiohi can survive infection from Plasmodium relictum. Our prevalence data supports that, and it indicates that Kaua'i elapaio may be even better at surviving the disease, as evidenced by that 30% prevalence. Both our prevalence and survival analysis support the hypothesis that honey creepers are particularly vulnerable to the disease. And while our small sample size of infected honey creepers prevented us from getting a really precise estimate of annual survival, we believe that this further supports the hypothesis of a high mortality rate for the group. Something to note is amongst all my survival models, all the way I did it, the highest annual survival estimate I could get was 74.8% for non-infected, non-honey creepers. This is lower than expected given what we know about the life history of these species. They're largely slow breeding and relatively long-lived for small species of birds, along with survival estimates for related species on other islands, which tend to sit in the mid 80% range. So why is even our highest survival estimate below 75%? There are two possible explanations. One is that the mosquito line is likely now at or above the highest elevation on Kaua'i. There is no mosquito-free habitat left there, while other islands, particularly Maui and the Big Island of Hawaii, do have some remaining mosquito-free habitat. This may depress the overall survival rate on the island of Kaua'i as compared to survival on other islands. The second is that our survival analysis couldn't account for undetected changes in infection status. It's highly likely that a lot of our uninfected birds became infected sometime after their initial blood test, and this would bring down the apparent survival for birds in the uninfected category. Future research ideas. So first, some honey creepers do appear to be able to survive infection of avian malaria pretty well, particularly Hawaii amakihi and apapane, the second of which actually survives it so well that it's become an important reservoir for the disease in some places. So further research encompassing a wider array of native species would be needed to determine whether honey creepers as a group are indeed more vulnerable, and these are exceptions to the rule, or whether it really comes down to species. Multi-state modeling in the survival analysis is a type of modeling that would allow for individuals to change categories, so in our case, infected versus uninfected, during the course of their encounter history. This would allow for greater precision in annual survival estimate. Unfortunately, we just didn't have enough detected status change in our samples to perform that kind of analysis. So of our over 600 re-encounters, just 83 were recaptures. Of those 83, less than 1%, 8 total, changed from negative to positive. And again, we didn't have any birds changing from positive to negative. Greater recapture effort of previously tested birds may generate enough data for this kind of analysis, but it's important to note that there will always likely be difficulties in getting enough recapture data for those birds who are infected and are particularly vulnerable to infection. Overall conclusions: climate change is causing the viable range of mosquitoes to expand upslope, and this is a huge threat for several endangered native birds on the island of Kauai. Some species may be able to adapt to the presence of avian malaria, but others will require significant mosquito control efforts if they are to be saved. And then while small sample sizes of infected akikiki and anianiao did confound our math a little bit, make it so we couldn't get a super precise survival estimate, this in and of itself is further evidence of the urgency of the problem. I want to thank my 3 committee members for helping me get through this process, particularly with, as Jeff said, all of the COVID obstacles that we ran into. I owe my life, liberty, and happiness to anybody who helped this field biologist get through the lab stuff and the statistical analysis stuff. That was, you can't imagine what a weight that was off my shoulders. If you're on this list, I really appreciate it. This thesis has been a collaboration with the Kaua'i Forest Bird Recovery Project. So I'd like to thank that organization as a whole, but there are also a few key staff members I'd like to thank. Justin and Tyler, members of the field team. They got me some great information and were able to answer some of my questions about the field work since I wasn't able to make it out there myself, as well as the lovely intern Hannah, who really helped me whip some of the data into shape. My final category, couldn't have done it without you. If you see your name on this list, then I am forever indebted to your moral support, your emotional support, everything that you did for me during this process. And I couldn't have done it without you. Thank you. Okay, I will take questions if anybody has any. Yeah, Kara.
Go back to slide 31.
Oh, the numbers, which, oh, here we go.
Okay. So I guess, like, I just have a question on your model. You say like high strength. I don't know.
Yeah, that's a good question. So my highest ranked model, what I mean is that I tried out a bunch and this was the best. A little more specifically, program mark, you put your encounter histories into it, and then you can start to manipulate some of the probabilities. So I could start with model and say, I think recapture probability is the same across the board, and I think survival probability is the same across the board. I'll put that in, it'll go and it'll spit out my estimates, and then it'll also give me a number that tells me how well my model explains the variance in my data. I use something called Ikake's information criterion. Then I go, okay, what if the recapture is different by species, but the survival is the same across the board? It goes, spits out my estimates as well as that goodness of fit number. And kept going, playing with it until I arrived on this best fit model, which had recapture different for all of the 4 quarters. It had recapture, which it mostly was recite, but recapture, recite probability different for each of the 4 quarters, as well as different for honey creepers and non-honey creepers. And then survival different for infected honey creepers and others. That gave me the best fit model. It tells me this is the model that does the best job explaining the variance in your data.
Just to follow up, also, out of curiosity, like, how many models?
I tried a lot.
Kelsey,
What?
can you repeat the questions? Because we on Zoom cannot hear the questions.
Oh yeah, you can't hear the question. Okay.
No, yeah,
So
please, thank you.
somebody, yeah, no, thank you for reminding me. Somebody asked about what highest rank model means, and then she just asked how many models I tried. I tried a lot starting from when I was teaching myself how to use the program. once I was really digging in the dirt of it, I tried, I think, a total of 12 models.
So
Yeah.
Tessa, and repeat this as well.
Oh,
Tessa has
yeah.
a question from online. Can you expand on the current or potential mosquito control efforts?
Oh, hi, Tessa. So mosquito control efforts, I have been told that there are some mosquito control efforts coming up on the island of Kauai that are going to be larger scale. I can't speak too much for the history of it. Callie might be able to jump in and answer something on that. But I know there have been some experimental looks at that.
Do you want me to jump in and answer that?
If you have the answer, I'd love that. Yeah.
Okay, so hi, everybody. I'm Callie. I'm the head of the Kauai Forest Bird Recovery Project, and I'm a member of Kelsey's committee. And before I jump in and answer that question, Kelsey, you did a fabulous job. Beautiful presentation. Thank you very much. So right now, we are all eagerly looking towards a mosquito birth control method that is based on fancy title, cytoplasmic incompatibility between males and females that have a certain bacteria strain or have a different strain of the same bacteria. So it's super complicated, but it turns out that most mosquitoes, if not all mosquitoes in Hawaii, contain, Culex mosquitoes in Hawaii contain a bacteria called Wolbachia, and it is involved in reproduction and successful development and fertilization of mosquito eggs. So mosquitoes lay eggs that turn into larvae and then hatch. So when males and females have different strains of this cytoplasmic bacteria Wolbachia, the male is not able to fertilize the female's eggs, and so therefore the female does not lay viable eggs. There is no offspring, and so it leads to population suppression. So currently what happens is all the males and females out there have the same strain of Wolbachia, and so there's no interference with fertilization of the eggs. All the eggs develop, we have mosquitoes. The idea is to raise males mosquitoes in the lab, inject them or give them a different strain, or find males that have a different strain of Wolbachia, rear them up in the lab in the thousands and tens of thousands and probably millions of mosquitoes. You can sex sort for males versus females. So you're only going to get males. You give them this different strain of Wolbachia and you release them on the landscape where they usurp copulations with females and sort of outcompete the males out there already that have the other strain of Wolbachia. And so the chances that a female encounters one of these lab-reared males with a different strain of Wolbachia are super high. So she has unviable eggs, population suppression. We are looking at trials of that on Maui starting later this year or early next year. On Kauai, we're slightly behind in the development of all of the environmental assessments and everything that have to happen and all the permitting and everything that has to happen. So we're looking at doing that on Kauai in 2024.
Awesome. Thank you, Callie.
Very long-winded answer, but it's kind of a complicated process. So, but yes, we are looking at landscape level mosquito control to change around this pathetic survival statistic that Kelsey has on her screen right now.
Awesome. Thank you. Hi, Phil.
Yeah, I actually have a comment and a question. I'll speak loud so you can hear me on the phone. I must be honest, okay, the idea of statistical analysis and that stuff is not normally in my realm of vocabulary, okay, but that, I actually understood that. So
I told you would understand it.
I understood it.
Thank you.
But my question is this, and it's more of a simple idea and answer. You said that you prioritize those captures to make sure to treat those that were stressed to do those tests first.
Yeah.
How do you know bird is stressed?
That's a good question. Basically, there's two ways. If they've got their bill open and they're panting, that could be a sign of heat stress or just stress. And then a really good way to tell is if your bird comes out of the little holding bag looking real sleepy. it looks drowsy and its like head is drooping and its eyes are kind of drooping, that bird's in bad shape, and you got to take care of it and release it.
Thank you.
Hi, Kelsey.
Hi.
My name's Kimberly. I'm a veterinarian here.
Oh, hi.
I reviewed the protocols, but I was very excited because I thought there'd be great pictures. And there were. Thank you. Those illustrations. Oh my gosh.
Oh, thank you.
I have a couple questions. So one is, what's the lifespan of the birds you were looking at, the honey?
The honeycreeper. So for what's that?
Repeat the question.
Oh, you're right.
Sorry.
No, that's okay. I just keep forgetting to repeat the question. So the question is, what is generally the lifespan for the species of interest that I was looking at? So for 3 of the species, they have all recorded life years of 10 years of lifespan of 10 years or more. Akikiki do not have currently a known expected lifespan, but we know that they do lay small clutches, which is in line with the longer lifespan, slower breeding strategy.
Thing I was thinking when you were discussing your sampling, what's the likelihood that you would sample a bird and not obtain a sample that had infected RBC?
Are you asking what's the likelihood that any given bird you catch it, you test it, and it's not infected?
No, so when you collect your blood sample and you run and you get your hematocrit, your red blood cell, what's the likelihood that a bird could be infected, that your sample didn't collect red blood cells that would have?
Oh, an undetected infection? That's a good question. I might have Jeff jump in on that. I know there are birds that can maintain a chronic lifelong infection that is often undetectable, but I can't speak to the specific likelihood of that. Jeff, do you know?
It was in correlation with your positive, negative, negative, positive. Could it be that you just didn't sample it at that time, or did it truly get infected after you recaptured? I got the plastic plate.
Yeah. So the question was, sorry, Jeff. The question was, in case anybody didn't hear or pick up from my response, what is the likelihood that you capture a bird that is in fact infected, but the infection does not show up when you test your blood sample?
So the answer is exceedingly low. It's not 0, but it's very unlikely that we'll miss it. But it's not certain. It's much better than any previous method, where previous methods were likely to miss, 10% or more of infections, especially at low level infections. But in general, we do not miss it, especially when we run it in triple pin.
And then one last question. Did you find any deceased birds that you have access to any that when you're in the field?
So unfortunately, I did not get to go to the field because of COVID. So the field methods were done by the lovely KFBRP field team. They don't attach, sometimes there are studies that try to find and collect deceased animals. This wasn't one of those studies. As far as I'm aware, they're not putting any transmitters or anything on these birds that would, put off a death signal if the bird stops moving for a certain amount of time. I'm sure that they have come across a dead bird during their sampling. I have not had access to that data, and it's not finding deceased animals is not an objective of the study.
I'll add that.
Yeah.
So I was there working for the same group in the 1990s, I'm aging myself. In the late 1990s, and we were starting to find dead birds in lower elevations that had died from malaria. So roughly 20 years ago, we started to see kind of avian malaria start to kind of move upslope, and we're beginning to find it just below these sites and more kind of middle elevation sites. So yes, dead birds are found. It's hard to find them, but you do find them from time to time.
Just in your discussion about what it might be, and that you weren't catching birds that have it because they're already deceased. I was wondering if you were able to see them, although they're small and probably get eaten by.
Yeah.
something.
It's possible that, a study could be done where if you catch a bird and it tests positive, you put a transmitter on it, or you try to recapture it as soon as possible and put a transmitter on it. But the field team out there is pretty small and they've got a lot of responsibilities. Kyle?
You just mentioned recapture, and there was a slide we talked about recapture versus
Recite?
recite. What was the difference again between recite and recapture?
So the question was, Jeff keeps coming to my rescue. The question was asking for clarification on the difference of recapture versus recite. So recapture means the bird hit your mist net, you've got it in your hand, you can take a blood sample from it. Recite means you're out in the field performing field duties, you spot a bird, you put your binoculars up, and you say, I can see the color bands, write this down. Somebody writes down the color bands, you take it back and you check it against the data sheet so you can find out what bird it was. So recite is you did not make physical contact with the bird. There was not an opportunity to take a blood sample, but you confirmed that it is alive because you saw it.
But for the modeling, it doesn't matter.
Right.
You're just confirming that it's alive again in a different period of time.
Yeah. So recaptures versus recites were treated just that you could use the word a re-encounter in the modeling. Yes.
My question is going to be pretty similar to Phil's, but early on you said that a symptom of avian malaria was anorexia. How does one diagnose a bird with anorexia?
So anorexia in people is obviously a pretty serious eating disorder. In animals, it just means it's not eating.
Okay.
Just means the animal is not seeking out foraging for and consuming food.
Is it just the behavioral aspect, or do you also like look at weight on the animal?
Both. So studies have been done that that 90% mortality for EEV that I mentioned earlier, that was done with captive birds. They had birds in captivity so they could keep an eye on them. The 90% mortality is sort of the hardest hitting statistic from that paper, but they did also measure serious body declines in other ways, including weight. So they had like one bird that made it, but realistically, if it was in the wild, it's not foraging, it's not eating, it's not moving around, it's vulnerable to predators. That bird would likely also not make it. So they can watch them if it's a captive study and see that they're not eating. They can also compare weight and see how it's dropping off. And birds have pretty quick metabolisms, so they lose weight quick if they're not eating. Hi, Hannah.
I have a question about the QPCR plot and your
Yeah.
lab methods. Sorry, I forgot to write it down.
The question was about this
Yes, that.
thing.
So you were saying that the QPCR plot tells you about like the amount of P realism in
Mhm.
each individual. You were saying the red line means it's more and the purple is less. How do you know that?
Yeah. So the question was asking for clarification on how to read this qPCR amplification plot. So PCR is a method of amplifying fragments of DNA. You put it in a machine that heats it, cools it on a very specific cycle with some primers, and it causes the DNA that's there to amplify so that it's at detectable levels. So you're not dealing with these tiny little trace amounts. What this shows us is, so the faster this curve picks up off that x-axis, that means that bird had a higher amount of that DNA in its system to begin with. If it takes longer, that means it is taking longer for the amplification process to create detectable levels of that DNA. So we know in this case, in this hypothetical, the red bird at the front started with the highest parasite load because it started with the highest amount of the parasite's DNA in its system. You can tell because that ramped up the quickest. This purple one is the slowest. It took the longest for the amplification process to make those DNA amounts detectable.
Actually, I have a follow-up question. So if there was no amount of P realism in the individual, then it would just not show up on the.
Correct. If there was none of the Plasmodium relictum DNA in there, wouldn't amplify, you wouldn't get a curve like this on the graph. Yeah. Kara.
I have a really quick question kind of on this topic. So you do a couple different things with your blood sample. How much blood sample do you need from each bird? And how was that transported so that it was still viable pieces of information from the lab site?
Yeah. So generally when you're bleeding birds, you take the bird in your hand and it's face up to you. You spread its little wing out and there's a vein, small vein. And I've seen Pete do this 100.5 times. You take something and you prick it, and then you take a capillary tube, which is a very thin, narrow glass tube. And all you have to do is touch it to that little blood droplet, and capillary action pulls the blood up into the tube. Tubes are about yay big and like a hair's width. So it's not a whole ton of blood that you have to take. And yeah, that was the question, right? How do they take it? How much?
Basically, you use it for, I think, I'm assuming like the amplification process is a separate thing than your electro.
Yeah,
So I would assume you need more than your.
It depends. Again, I'm a field scientist who had a lot of help in my lab methods, but that's part of what amplification is for as well, right? Is if you have your sample, you don't have to use the whole sample to have detectable amounts of DNA. You can use a part of it and use PCR to amplify it. Yeah.
First of all, those illustrations at the beginning, again, those are fantastic.
Thank you.
Something good is happening with those, or selling them or something, but.
I was going to
That's
gift
good
them to
stuff.
KFBRP.
Right, those are good.
Thank
Yeah.
you.
I was hoping you could maybe tell me a little more about the evidence to suggest the high mortality in the honey. I can't recall like the population estimates, like are the honey creepers declining faster than Elapio too? Or were there like lab experiments to show that high mortality? I'm kind of curious where you're getting the high mortality from.
Yeah, so the high mortality estimate, yes, good Lord, I'm terrible at that. So Pete asked for a clarification on where my declaration of a high mortality rate for the honey creepers is coming from. So it's coming from a few different places. Here, we'll pull up this. So first of all, there's prevalence, right? So what we know about prevalence is each bird had to be well enough and active enough to be flying around to get caught in a mist net. If you remember at the beginning, there's something like, I think great tits had a 91% prevalence in one study. That means that 91% of those birds have avian malaria and are still flying into mist nets. So that tells us that those birds handle it pretty well. So on the opposite side, when you see birds with such a low prevalence rate, especially in an area where you know that avian malaria is present, that kind of tells you that it's not that they're immune, it's not that they're not catching it, it's just that the likelihood of catching one of those birds is very small because it's going to get very, very sick. It's going to stop foraging. It's not going to be flying around. It's not going to hit your mist net, and then it's going to die. So there's that. And then we also have our survival estimate here. So this is based on, basically it'll look at the encounter history, and you might have saw it, didn't see it, didn't see it, didn't see it, saw it, didn't see it, saw it. And the computer will use those periods of time where you didn't see it to calculate, if you stopped seeing a bird, what's the likelihood that it's because it died versus because you just didn't see it during that particular sampling period. So it uses that to calculate that likelihood. And so this is telling us that infected honey creepers, by its estimate, they are dying. You know, and I can say just from looking at my own raw data from endless spreadsheets and putting this stuff into the right format, for our Kaua'i Elapio, you'll have a Kaua'i Elapio that tests positive the first time and you see it 6 more times. That's not the case for any of our infected honey creepers. They'd get a positive test and you'd see them maybe 1 more time and then they're gone. Yeah, thanks. Alex?
I believe you mentioned that in comparison to the native birds and the introduced species, that the the migratory birds seem to be relatively unaffected by this particular strand of avian malaria. I was just wondering why that might be.
So you are referring to this, yes?
Yeah.
So for a while, people thought that this malaria might have been introduced by migratory species, because there are migratory species of birds that stop on Hawaii that can and do carry avian malaria. The thought was just there were never mosquitoes before. So there was never a vector to transfer it from one bird to the next. So there are older papers, like from the 1960s through the 1980s, that declare that this is likely where it came from. However, once this particular strain was isolated and it was found that every single case of avian malaria on the Hawaiian Islands was this strain, they could look at where it came from. So they compared the strains found in those migratory species with the strains found in the non-native introduced species, the more terrestrial forest species, and they found that this strain just really wasn't showing up in those migratory birds, and it was pretty common in the non-native introduced ones.
All right, if there are no more questions, we'll wrap it up and give a round of applause for Kelsey.
I don't know how to pull the Zoom thing back up. You're right, Kara, you're so smart. Thanks, everyone. Kelly, it was so sweet of you to show up. Thank you. Steph, hi. Okay.
You could phone raise your next